thank you all for your presence here. So today we are happy to welcome Lijing Xin from EPFL. So for this 70 seminar. So Lijing will present us her work about spectroscopy. So Lijing is a research staff scientist at CIBM MRI EPFL, and she's also assistant professor at EPFL. And thank you again, Lijing, for accepting this invitation and to present your work about 7 Tesla spectroscopy.
Thank you for the kind invitation and introduction. I will continue the topic, stay on the spectroscopy from the last series, and but more towards metabolism and ex-nuclear MRIs in this talk. And I have to say, this is not one person's work; it's a teamwork from all the people working at EPFL 70 group. So we are interested in MR spectroscopy because it's a modality provides us the information about molecular information, not only restricted to water, as we usually see in MR imaging, provides the properties of water, give us structure and functional information. But we are looking at much smaller scales where you see the foot of the water there, a lot of hidden information related to many metabolic information like energy metabolites, neurotransmissions, membrane metabolism, and antioxidant or osmolytes. And to extract as much as you can, the information need state-of-art technologies and measure those metabolites reliably. And not only static information, which we can measure, like the level of metabolites in the brain or in different organs. It's also important to know MRIs can measure dynamic informations. This example showing the metabolism of energy, where you can using multi-nuclear MRIs measurement to extract the metabolic information related to mitochondrial function like TCA cycle metabolic connectivity activities, and also the ATP synthesis activities. And all this kinetic information, which is very important, provides information related to neurological disease and brain functions. So to measure those ex-nuclear MRIs is very challenging because these nuclear have rather lower sensitivity. Here you can see the example of MR imaging sensitivities, like the peak height of the mountains, and you can see the very low sensitivities for phosphorus is the height of the ends. So they are like magnitude of order of differences, and makes the measurement very challenging. And this requires the application at ultra high field, which boosts the sensitivity, and also need cutting edge hardware, especially like IF coils. And this will be covered by Daniel in September. And then we also need efficient acquisition methodologies to boost the sensitivities. So in this talk, I will cover mainly focus on the carbon-13 phosphorus and proton MRIs. I will cover some localization methods and focus mainly on the GABA detections, the single voxel measurement, and spatial mapping of GABA, and also application studies. Then we go to the phosphorus MRIs to introduce you a bit what we can measure from phosphorus, and some applications related to phosphorus MRIs, and also a fast measurement for kinetic measurements related to ATP metabolism using phosphor MR fingerprinting. At the end is the C13 MRIs part, which I will introduce you how actually we can use C13 MRIs to monitor energy metabolism, to study brain metabolism, and the application in not in human, but in an animal model, and also the setup we have for the 70 human experiment. So localized MRI sequence at ultra high field, usually we want to do it at short TE to keep the sensitivity as much as we can. And here is the custom sequence, which reaches a short TE for single voxel MRS. It's a special sequence based on ISIS localization and a double spin echo sequence based on adiabatic pulse. And that allows you to actually reduce the chemical shift displacement relative to a product price sequence. As you can see here on the right side is the adiabatic pulse actually align very well for the intended excitation slice between the two metabolites. And also we are very interested in doing ultra-fast spatial mapping for metabolites. Often now going faster, we have to go to non-Cartesian spatial encoding. This is the case for the rosette trajectory, which you can reach ultra short T with 0.83 milliseconds, and you can actually reach the metabolic mapping of major metabolites. But what sometimes we are very interested in those low concentrated metabolites, especially for instance, GABA actually caught attention for many research projects. This is inhibitory neurotransmitters and a very key molecule in our brain, facilitates many neurological processes. And it's very difficult to measure because it has very low concentration, about 0.5 to 1 millimolar, and it has strong overlap with the different metabolites on top of it, especially creatine, which is very high, resonating at 3 ppm. And we say, okay, often we go to 7 Tesla, we have improved this spectral resolution. As you see, the bottom left here, people usually demonstrate at 70, you have a separate glutamate and the glutamine detection. You can see the peak around the 2.3, 2.4 there, you have a separation of glutamate. And on the right side, you see the fitting of glutamate glutamine at 70, indeed actually have a separation where you actually facilitated the separate measurement. But if you look at highlighted GABA actually underneath of many metabolites, you were not able to resolve the peak from that and make its measurement very challenging. So we say now we go to the high field, the 70. Could we still measure, GABA using short T rather using editing methods, which we targeted specifically for GABA? Is the short T sufficient enough to measure it reliably? So with Song Yi, so she implemented a mega special sequence. It's a single voxel editing sequence specific for GABA, using a adiabatic pulse to increase the editing efficiency, make it insensitive to the B1 homogeneity and the B0 offset. And then there's an additional Gaussian pulse to remove the contamination of the co-edited macromolecule at the same resonances. And you can see here on the right side, there's edited GABA peak resolved from the intensive peak from total creatine on top of it. And but you get also glutamate on the left side. So then she did in 2 brain regions a retest study to see, especially when we have the question, you apply for longitudinal experiment, we want to see actually if you put the subject multiple times, how reproducible is your result. And especially for the GABA measurement, that also tells you how sensitive is your methodology. And we see that with the solid circle is the short echo time measurement, open circle is the editing methods. And here there's 4 dots representing different acquisition time with a number of averages. And you can see clearly in the M1 region, the motor cortex region, you can see the short echo time actually has really bad reproducibility, reflecting by high coefficient of variance between test and retest studies. Well, if you look at the mega editing GABA methods, it's remain much lower coefficient of variance and much more reproducible. And when you do 128 averages, you will be able to make the coefficient of variance below than 5%. While in the MPFC, where you have higher GABA levels, you can see the two methods actually works equally well. And with the editing method, you can see if you run at 32 averages, which is basically 2 minutes of acquisition, you can measure GABA reliably in both regions in the M1 motor cortex and the prefrontal cortex. So we say the editing method indeed actually can provide a more reliable GABA measurement at 70 compared to the short echo time, especially in the regions where you have a low GABA concentrations. As you know, the neurotransmitter is not distributing equally across the brain. And then we apply these methods to an application study where we're looking at in elderly subjects before and after a physical exercise training. In the case here is a balance training, which is supposed to promote the inhibition in the motor cortex. And we applied both short echo time methods and the GABA editing methods. And you can see only the GABA editing method capture the changes, the increase of GABA after the balance learning. So again, showing the GABA editing method in the motor cortex actually has a better sensitivity to detect changes, especially longitudinal changes. And then that comes the work from Andre. He recently implemented the GABA mapping using a slow editing sequence developed in Bern, and applying this together with a recent spatial encoding with rosette trajectory. And you can see also nicely the resolved GABA peak in the demonstrated grid on the bottom left, and the map of GABA in the slides where it's shown on the bottom right. So that's able actually to measure now the spatial distribution of GABA and apply them to longitudinal studies, which might be more actually sensitive tools to capture changes introduced by interventions or disease progression. Another thing I want to mention is we can also use MRIs to study brain functions, as we're very familiar with fMRI, which you measure the hemodynamic changes were actually coming from the property of the water in the blood, where you can see the property of the water molecular changes and reduce the bold signal, reflecting mainly the blood flow changes and the oxygenation changes. While in the MRIs, we apply similarly dynamically MRIs measurement during the functional paradigm. Here is an example of visual stimulation where we measure MRIs dynamically during the visual checkboard task. You can see there's a change during the visual stimulation where you have increased glutamate and lactate. And this reflects basically during neural activities, you need to actually have transient energy support coming from, for instance, glycolysis, where you see it reflecting in the lactate changes. And on the other side, there's increase of oxidative metabolism to support further energy. And you see the glutamate changes actually generated from a TCA cycle in the energy metabolism. So now we go to the phosphorus MRIs. It's very different spectral feature as what we usually see for proton MRIs. There's peaks at different chemical shifts, and they represent different groups. Some are involved in energy metabolisms, for instance, like ATP, phosphocreatine, inorganic phosphate, those are famous metabolizing energy metabolism. There's also important metabolites involved in membrane synthesis and degradation. They are often changed during, for instance, if you have brain tumors, these membrane-related metabolites, they will substantially change. And also we can monitor the intracellular, extracellular pH, also the magnesium concentration. And recently, there's a more interest in this small peak on the right side of the alpha ATP, where you have the NAD plus NADH, which the two redox pair involved in energy metabolism. Of course, there are also one coenzyme in co-substrates in some cellular signaling pathway. And we are actually looking at, for instance, this one application study performed by collaborator in Harvard. They look at NAD plus NADH in patients with schizophrenia and also bipolar disorders. And you can see an increase of NADH and the reduction of NAD plus NADH ratios. And this is an indication of a mitochondrial dysfunctions led by redox dysregulation. So another collaboration with industry, Nestle Health Science in this case, we performed a clinical trial with a nutrition product. It contains the medium chain triglyceride, which you can boost the ketone levels in the blood. And in this case, which you can measure now in the brain, the NAD plus NADH by uptake of MCT, increase of ketone bodies in the brain, and facilitate the energy substrate taking from ketones actually can be more efficient to produce energy and reduce the NADH and improve NAD plus and leading to an increase of NAD plus NADH ratio. And this can be a potential strategy to against, for instance, aging, neurodegenerative disease or psychiatric disorders often has a reduction of this redox pair. And for doing the phosphorus MRIs, often people use a surface coil directive for localization. In most cases, it will be the occipital lobe. And if you want to do a sequence localization, you can see on the top three is the common sequence localization sequence we use for proton, but not work properly for phosphorus. And the bottom one is the IC sequence is based on the inversion and the FID sequence that works much better in terms of sensitivity for phosphorus. The reason for that is the relaxation property of phosphorus metabolites. They usually have a much shorter T2 compared to the proton one, especially the ATP, which has quite short T2. And using ICIS, which has a short T measurement, you have a substantially boost in ATP signal relative for other echo-based methods like semi-laser, steam, or especially where you have echo time on the order of like 10 to 30 milliseconds, for example, and the ICIS will have just 2 or 3 milliseconds. And then Antonia actually conducted a study combined with functional MRIs using phosphorus and to look at actually how the NAD dynamics changing during brain stimulation in the case of visual stimulation. She used the 3D IC sequence localized in the activated areas in the occipital lobe. And applying measurement dynamically, she observed for the first time the reduction of NAD plus in the first stimulation areas, but not the second following stimulation areas. This means the NAD has a reduction support first stimulation of the brain to support the energy which the brain needs, while in the consequent upcoming stimulations, maybe the energy provided by oxidative metabolism catch up, which you do not actually need NAD plus reduction to support for energy in the following stimulation. Of course, we are also interested in the whole brain information about NAD. This is one a straightforward application using CSI FID to map the NAD and other phosphorus metabolites in the brain. And this was conducted by a 32-channel phase array coil from phosphorus with a high sensitivity gain, and we were able to successfully to report the NAD map from the brain. Here is one example from other groups. It's an application of the phosphorus mapping, including NAD in gliomas, where you can see the left side is astrocytoma and the right side is the glioblastoma. There's a different metabolic feature in terms of phosphorus metabolites, where it can provide information about tumor metabolism. And there's another interesting technology in phosphorus mapping is the ROSA trajectory, which we use also in the proton. In the phosphorus, there's a ultra short TE implementation with only 67 microsecond in this case, where you can see on the bottom left side, you see compared to the conventional FID with the longer echo time, you can see on the left side with the rosette, you have a gain of SNR with this ultra short TE. And also, especially you can see the ATP signal on the right side where there's 3 little bumps there. And this application, they applied at 3T, where they can further accelerate the measurement using compression sensing with up to acceleration factor of 4. Not only measuring the static information, for example, we map the phosphorus metabolites, but we are also more interested in kinetic information like the enzyme activities involved in ATP metabolism. ATP is synthesized in electrotransport chain, which involve an important enzyme called ATP synthase. And there's another enzyme actually can save the energy in the phosphocreatine and move it to a place where you have energy consumptions during your normal brain function. And this is called the creatine kinase enzyme, which store the energy and move it there and release the energy. So these ATP synthase and creatine kinase are two important enzymes involved in ATP metabolism and transportation. Why we are interested in enzyme activity instead of just measure the ATP or phosphorus or inorganic phosphorus levels, because this is one example where you can see on the top where you have the ATP levels across different brain activity measured by EEG from the left to the right. The left is the lowest activity and the right side is high activities, where you see the ATP has no change, highly stable, highly regulated in the brain, stays at 3 millimole, for instance. While if you look at the ATP synthase metabolic flux, you can see actually it's lower and correlate with a brain activity stage. And there's another application in schizophrenia patients prove the similar concept. If you measure, for instance, in patients with ATP or PCR, PI levels, they remain quite stable and unchanged, especially for those diseases which are not very aggressive, and you see no changes at all here, but you already can actually capture a creatine kinase activity changes in the early stage of schizophrenia, which makes this enzyme activity more attractive as they can actually show early changes before you see the metabolic resting states level changes. And to measure this ATP metabolism is quite, let's say, complicated compared to the rest measurement, as it involves the manipulation of magnetization using magnetization transfer experiment. If we just measure the levels, even many chemical exchange happens during the living processes, but you can see the level of ATP phosphocreatine in organic phosphate, they remain the same. But if we start to introduce magnetization transfer to saturate the signal, for instance, in the ATP here, and by doing the chemical exchange, this saturated signal going to the neighboring molecule, which has chemical exchange with the maple. And with more the chemical exchange, you will see the more reduction of the signal on the molecule which has chemical exchange with the ATP. In this case, we see the reduction of the PCR and the PI with the time by keeping saturating the ATP signal. Then by looking at how fast the signal decays, that's reflecting how actually fast the kinetic information of ATP synthase and creatine kinase. So doing the mathematical modeling, you can extract the metabolic rate of creatine kinase or ATP synthase. As you can see here, we need a multiple measurements with different saturations. So this also has to be performed with a rather long relaxation recovery time, which lead to a long acquisition time, also give you a very low temporal SNR. And also we have to do magnetization transfer, which you have to apply for saturation pulses. That give you also high energy depositions. In the case either not able to run this at 70 or just give you a super long acquisition time in terms of TR. And so all these classical magnetization transfer approach, they are very time consuming, and that's also not very feasible, I mean, for the spatial encoding to do a global measurement over the brain. So we actually come up with a solution called phosphorus MR fingerprinting to deal with these issues and to bring a more efficient sampling scheme for measure the kinetic changes in the brain. And also we target the B1, especially the B1 in homogeneity, which have often demonstrated at 7 Tesla. We will also implement a method to measure the B1 of phosphorus at 70. So for those not familiar, what is magnetic resonance fingerprinting? It was a method proposed for imaging more than 10 years ago. And the idea is to using balanced steady state free processing sequence. And this sequence is highly SNR efficient, but it's also very sensitive to T1, T2, and for instance, off resonance or even B1. And by changing different acquisition parameters, for instance, TR flip angles, you are able to reach a different signal patterns according to their differences in T1, T2, and off resonance, for instance. And this unique pattern can be predicted by simulation with a large library. And by matching the pattern from the measured one and the one in the simulation, you will be able to find the best match and to derive the parameters and to do the mapping, for instance, for relaxation, T1, T2, and B0, for instance. So we want to adopt this method because it has high SNR efficiency, which is what we need in the phosphorus. And also it provides a multi-parameter estimation, which we need also in the phosphorus measurement to measure the kinetic rate, also the relaxation properties often linked to the kinetic rate in phosphorus. And the most important thing is it doesn't need an inversion pulse, which can introduce the inversion transfer and allow us to reduce the SAR if we use a saturation transfer instead. So here the inversion transfer can also substantially reduce the energy deposition and allow us to run this at 70 fast. So the whole framework, the sequence is on the left side, which you can see there's a two selective inversion to invert PCR and the ATP alternating. And then you can see the blue and orange shows the fingerprint signal in the bottom where from the PCR and the ATP. And then you can see on the right where we synthesize the large library with different parameters and generate different signal patterns of PCR and ATP. And by finding the best match between the signal pattern we acquired and the one we simulated, we were able to derive multiple parameters out of that. And what you can see here, there's many parameters can affect the signal evolution. For instance, the creatine kinase activities and relaxation time of T1, relaxation time of ATP, PCR, and also B1, B0 inhomogeneity and the concentration of ATP and PCR. So that's actually make a challenge in the data processing, because we accelerate the acquisition, and the acquisition can go down now to almost 20 times faster, but we have more complicated things to deal with, is our signal is sensitive to many different parameters. We have to disentangle them from the signals. And by creating a library with many parameters, it's very time consuming. It's not feasible with current computed settings. So Mark, in this case, developed a nested iteration interpolation methods where you can split the problem to linear fitting and doing the interpolation to correct the bias by the linear fitting. We will not go to the detail here, but can talk about this later if you're interested. So this is technology which he developed and patented for dealing with large dictionary fittings to achieve parameter in MR fingerprinting. And then they measured this proof of concept in one slice measurement in the occipital lobe, where you can see the KCK measurement and the T1 measurement of PCR and the ATP, where the blue one is the MRI result and the red one is the traditional inversion transfer methods, and they actually the value corresponding very well to the traditional methods. And then we also did a test-retest coefficient of variance measurement. You can see the blue ones are still the MRF, and the red one is the Abbott method, the traditional methods. Using MRF, you can reach much higher, much better coefficient of variance, even at a very low SNR with acquisition time of 4 minutes. You can reach the same coefficient of variance of a traditional methods measured at 17 minutes. And using a saturation transfer, our previous study shows the coefficient of variance is approaching to 20%. With MRF, you just use 2 minutes, you can reach 20%, even below about 20% in the current setup. While the saturation transfer will need almost 50 minutes to measure that. So the time reduction is very impressive here. So now he extended this method from one slice to the 3D measurement using the spiral trajectory to map the PCR and the ATP using frequency selective pulses. And this is a demonstration results where he obtained with 21 minutes, in the brain. He obtained a KCK map across the whole slice in the coronal view here, and you can see the value, mean value, which is showing the bottom, is in the range of the value usually reported in the literature for creatine kinase kinetic rate in the occipital lobe, for instance. So the value is also in the range of reported one in the literature. And this is, in fact, the first report for the 3D KCK map in the brain. And he will present this work in the ISMARM in 2 weeks, which if you go to ISMARM, you can actually listen to his talk. He will talk more about these methods there. And since we have all these parameters also be affected by B1 at 70, with Mark, he implemented additional methods using spiral trajectory and weighted average double angle methods to achieve fast phosphorus B1 mapping methods. Maybe sounds not very interesting here for those which measure proton B1, because it's pretty fast, like less than a few seconds, you can get a B1 map in proton, but it's not that straightforward in phosphorus. As I showed at the beginning, the phosphorus is like ants, right? The proton is like the mountain peaks. So the sensitivity is already much, much worse compared to protons. And so here the measurement, usually if you do a double angle method for phosphorus without a cutting edge coil, it's for single subject measurement, it's not feasible at all. And here using a 30-channel phase array coil using a traditional double angle methods, you can measure it around 26 half an hour. And using these fast measurements, you can do it with 11 minutes. So this you can incorporate in the single subject measurement and to correct the B1 inhomogeneity in your data sets. Okay, I just want quickly to talk about the last session is the C13 MRS. It's a tool we measure brain energy metabolism. And what we look at here on the top, where is the C13, where you cannot see any, almost nothing like in the bars, because just has very low sensitivity, also very low natural abundance, which means in your bodies, you have only 1.1% carbon is carbon 13, and the rest is carbon 12, which is not visible in NMR, right? And so this seems very bad drawback for carbon 13, right? But actually, this is something we can take advantage of it because it doesn't exist too much in the body. So we can introduce exogenous tracer and to monitor the C13 metabolism with that. So often for C13 MRIs, we apply C13 labeled substrates like glucose or acetate or lactate, where you can do it by oral uptake or intravenous infusion. And when these C13 labeled energy substrates across the blood-brain barrier, and they will enter the energy metabolic pathways. And then you can actually monitor them by C13 MRIs. In the case, we can only observe here, for instance, for those shaded color in blue, like a glutamate, glutamine, lactate, glucose, those actually reach the sensitivity detection level of MRIs. We cannot see pyruvate or acetyl-CoA because the concentration of them is just too low. They do not fall into the detection threshold of MRIs. But already looking at the glutamate, glutamine, and lactate, how the signal evolution across the time, where we can see here the signal of, for instance, glutamate C4 position grows, and also the position C4 of glutamine grows, then we can see the change of the C13 signal entering the glutamate and the glutamine. And again, if you remember what we talked about before for phosphorus magnetization transfer, here we are using exogenous tracer and looking at how fast it's entering into the end products of the TCA cycle, where is here is the glutamate and glutamine. And this actually reflects how fast the TCA cycle running, either in the neuronal compartment or glial compartment. And also it can tell us how fast is the glutamate and the glutamine cycle between the neurons and the astrocytes. That gives us additional information about neurotransmission. And these are all dynamic measurements instead of what conventionally we measure, for instance, glutamate and the glutamine level in proton spectroscopy, where you only have a resting states concentration level. Here, it really provides you dynamic information about the energy metabolism in the brain. And often we will say glutamate levels is low in the brain, but this is not necessarily saying, okay, your glutamate tertiary neurotransmission is low, but using C13 MRIs, you may able to answer this question. And here is a modeling from the 3 compartment, the GABAergic neuron, glutamatergic neuron, and the glia, where you can measure glutamate, glutamine, GABA, all these metabolites, which we can observe by C13, and we can extract the TCA cycle metabolic rate in 3 different compartments, and also provides us information about GABAergic neurotransmission, glutamatergic neurotransmissions. One example I want to show here is we apply this in animal model for study schizophrenia. And you can see the hypothesis in schizophrenia, which involved in the redox dysregulation and glutamatergic system problems. And using C13, we want to target this. So we use an animal model with redox dysregulation, which has a low glutathione level. Glutathione is an antioxidant in the brain. The low level of glutathione in this animal model lead to actually characteristic features of schizophrenia, and it has impaired GABAergic interneurons, and this is very much mimicking in the post-mortem observation in patient, and it also has abnormal myelination behavior status. And we can see here using C13 MRS, we were able to measure the TCA cycle activities in both neuronal and astrocytes activities. And we can see there's a reduced glutamate turnover in both astrocytes and neurons. And later on, it's more interesting by looking at the metabolic flux between the measurement from C13 and the measurement from protein levels. We can see the GAD67, which is a GABA synthesis enzyme, in the red, which is the white type, where we can see the neurotransmission flux is negatively correlated with GABA synthesis enzyme in the white type, where when you have a low GABA synthesis, you have higher activity in neurotransmissions in white type. While you have a higher inhibition, higher GABA synthesis enzyme, you have a lower neurotransmission, which makes sense in the excitation and inhibition balance. While if you look at the black dots from the knockout, you have a lower the GABA synthesis enzyme, you will see actually a deficit in the neurotransmission there. So this is already shows the neurotransmission is dysregulated in those which has a low GABA activities. And that's not only show with the neurotransmission between glutamate and glutamine, it also shows the neuronal TCA cycle activities. The plot on the right shows the same issues. So there's a dysregulation between GABA and the glutamatergic activities, both in neuronal activities, also in neurotransmission. To translate this into human setup, we prepared a C13 coil for the frontal lobe, which is often people are interested in study cognition or psychiatric disorders. And the sequence was also implemented for the frontal lobe to be able to monitor the C13 MRIs using ice-stepped polarization transfer sequence to enhance the detection of carbon-13. Here is one demonstration of C13 myoinositol natural abundance signal in the frontal lobe. Here we show a natural abundance. Think about this 1.1% of the signal here. And doing a C13 measurement is very attractive because you measure real neurotransmission and TCA cycle activity, but it's also very costly because of high cost of carbon-13 substrates like C13 labeled glucose. So come to the end, I want to make a summary here, which today I talk about the protons, the phosphorus, the carbon-13, the nuclear, but not only limited to these nuclear, there's other nuclears like deuterium, are also very interesting, sodium imaging as well. And all these X-nuclear imaging techniques provide us molecular information beyond water molecule. And this information, not only static information, also dynamic information, provides us the underlying molecular mechanism of brain function and pathologies. But to measure those information is very challenging. We use ultra high field, the 7 Tesla, which is very great for sensitivities, but there's also coming with limited challenges, limited resolution, or also the scanning time due to the site depositions in the methods and lead to long acquisition time. So if we tackle these challenges and use more efficient acquisition and analysis strategies, we can bring the value of multinuclear MRIs to the clinical setting to answer clinical question underlying the pathologies. And that actually will provide us a translational hub to explain what we see in the molecular neurobiology in the animal side, and translate this to the human side to provide information in cognitive neuroscience or clinical research. And then I would like to thank all the team members at 7 Tesla and also collaborators from the CIBM partner institutions, the Siemens team as well, for strong support of the 7T development, and also external collaborators from industry and institutions abroad. Thank you for your attention.